Very good morning, students. So today we will be starting with one of the very beautiful chapter, that is regarding the complete part. Yes, that is regarding the complete part, that is the reproduction in flowering plant, students. So today we are starting with that part. I hope I am audible to every one of you. I hope I am audible to every one of you, students, right? Okay. So great, students. Just let me connect to the YouTube through my mobile so that I can see you people. Just give me a moment, students. Okay, so even fine. Am I audible to every one of you? I hope my voice is audible to all of you, students. Just let me know if I am audible. Okay, great. Okay, you can chat with me, students. In the chat box, definitely you people can chat with me. So now, so shall we start? Okay. Just try to connect the laptop. Yes, it's done. All is set now. Now we can start. You see, we will start with the part now. Let us go ahead. So what do we see? We are going to learn the reproduction in flowering plant, angiosperms. That is one of the very important content which we are going to complete today. One of the very important topic. Now, what do you see over here? That is, just let me make a little setting, student. Just give me a moment. That's good. Okay. Now I can definitely talk to you all as well as okay, students, shall we start now? Now, in this particular chapter, some of the important part which we are going to cover and which are very important to the learned student, that what all we can see. So pollination and outbreeding devices we are going to do with that, as well as we have got one more, that is the sexual reproduction in the flowering plant. In the flowering plant. So we'll start with that. Okay, shall we start? Okay, good. So when we go to the sexual reproduction in flowering part, student, you can easily see that is these all organisms. If you see, reproduction is not only one of the property of only plants, it's also the property of animals also, which we have done earlier, that is human reproduction we have done. So altogether it is going to contribute towards the progeny to be prevailing in the next generation. So this comes out to be one of the very important aspect through which we can learn about the organism and their complete population, how it is going to be working. Now flowers, if you see as such, it has got two particular bases. Flower, if you see, it has got two particular bases, students. One is said to be as such to be non-essential parts, and the other comes out to be is essential parts. The non-essential part comes out to be sepals, as you can see over here. So it comes out to be sepals, right over here you can see, and the petals also, because they do not contribute anything towards the reproduction in the organism. These all portions are not going to contribute about anything in the reproduction we are going to see. That is one very important factor which we are able to understand. Whereas when you talk about the essential part, it comes out to be androecium, that comes out to the male reproductive organs, and the second comes out to be is the gynoecium, which is the female reproductive part, which is going to be a female reproductive part in the organism, students. So that is also one very important factor which we are going to notice. Now gynoecium is also called as carpel. Okay, gynoecium will be also called as carpel, as well as we also call it as pistil. Androecium we also call it as stamen. Androecium we also call it as stamen. So these are the different names which has been assigned, which has been given to the complete, that is the structures. These are the complete names which has been assigned and given to the structures, as you can see, students. Right? Have all of you understood? Now just give me a moment, I want to just see once if any student, yeah. Okay, give me a moment, students, just let me see if I can. Okay, I think it is, okay, shall we start now? I wanted to write over this also if it is possible, so that it will be more be accessible to all of you if writing was possible. If I could use my pen tab and we can write it over here, because this will be really helpful for all of you. If writing was possible, I could have written. Okay, if possible, let me see if I can write over the clip art. It will be easy for all of you. Something could be written over here. think that part is okay. I think now this is more better visible to all of you, and you can more better understand this particular part, right? If you see, so we are done with this part, so you can see over here, yeah, I think this is more better, definitely. Correct? So we see where we have got the flowers, okay? The flowers are there, as well as from the flowers we can see these are the non-essential part, they are the essential parts we can see. Sepals, petals, already we have done with this particular thing, sepal, petals and everything we have already done, student, right? Okay. Along with that, what next we are going to see, yes. When we go to the structures of the complete flower, now we'll go with the structure of the complete flower. You can see the flower has got the male reproductive part as well as the female reproductive part. As you can easily notice over here, it has got the male reproductive part and the female reproductive part. The carpel, it consists of we easily can conclude that so the male flower is right over here, you can see, and then we have got the stigma. Now the male flower, when you talk about the male flower, it can be as easily understood that the male flower consists of, the male flower is going to consist of some of the parts which we are going to look into picture, okay. The complete, the anthers and the filament, as you can see over here, we have got the connective tissue. Here we have got the connective tissue, then we have got the anther and the microsporangium, as well as the stamen is present. When we go to the female part of the structure, it consists of stigma, style and filament. Stigma, style and filament, these are some of the structures which you can see. Another is the part that is the ovary part. In the ovary part, now in the ovary part, if you notice, there are ovary which consist of ovule, and it consists of ovule, student, okay. Now in the ovary you can see there are presence of ovule. So ovule are the the female reproductive structures which are further going to help in the formation of, they are further going to help in the formation of the complete, that is the seeds which are going to be, and the ovary will be developing into the fruits. They can be developing into the fruit, students. That is one very basic fact which we can notice over here. They are going to develop into the fruit, students. Now what do we see? There are nectaries also present. Now these nectaries which are present in the plants, they are basically responsible for the nectaries which are present in the plant. If you see the structure right over here, if my cursor is visible to every one of you, the nectaries basically they help in the complete extraction of the sugary material, and in the search of it many of the flies are going to come nearby, and they will be contributing towards the complete, that is formation of the exactly the nectar through which the insects will be, pollinating insects will be attracting toward it, and it will be helpful in the pollination of the plant. Similarly, we have got the floral axis also. Floral axis also, as you can see, the floral axis is present on which sepals, that is the calyx part, corolla, that is the petal, androecium, that is the male reproductive part, anther, and the gynoecium, that is the pistil part. So 4 whorls are present. We generally say there are 4 whorls which are present. That is one very important factor, students, which we are going to look into. That is how we are going to see. So I hope everyone has understood this particular part, student. Okay. So this complete structure, I guess, is understood. There is another structure that is called as perianth. When we talk about the perianth, student, it can be easily understood that this perianth structure, the perianth structure consists of the corolla as well as the calyx together. We call it as a perianth. Petal and sepals. Petal is the corolla, sepal is the calyx. So perianth is nothing but the corolla and the calyx which are consisting together. That is how we are going to find. Have you all understood this part? Till here, is it understood to every one of you? Now we will go to the next structure. Let us try to see. Sexual reproduction in flowering plant we will talk about. What I was talking, androecium, gynoecium in detail we are going to do right now. So you see, when you see the complete structure of androecium, you see it consists of the anther and the filament. These are the 2 external morphological structure we are going to find in case of the, these are the 2 morphological structure which we are going to find in case of the complete androecium part. Okay. And there is a transverse section TS given to the androecium. There is a transverse section which has been given to the androecium part, and let us try to understand how it is going to be. Let us try to understand how it is going to be and how it will be working. Okay. Shall we go ahead? Shall we look into that part? Okay. So when a transverse section is given, student, to this androecium part, we find that it is a 4-lobed structure to be so, and the spore sac can be present. Okay. Pollen sac or spore sacs are present. So we call this structure to be a tetrasporangiate condition. We call it as a tetrasporangiate condition. Okay. Have all of you understood this part? We call it as a tetrasporangiate condition. That is one very important factor which we are going to see. And now, yes, very good evening. Yes, very good morning. That is Akshay. Akshay, very good morning. Now, we also find that in the tetrasporangiate condition, 4 pollen sacs are present right over here. 1, 2, 3 and 4. 4 pollen sacs are present which are filled with the pollen grain. They are going to be filled with the pollen grain, student, which we can see. So this is how it is present. Then we have got the connective tissue in the center. You can see these are the connective tissue, and these corners, these margins are said to be the dehiscence point. We call it as a dehiscence point. Why do we call it as a dehiscence point, student? Because these are the point from where we find that it is going to dehisce, or rather I can say it is going to break down. It is going to dehisce or break down, student. That is one very important thing which we are going to see. They are going to dehisce or break down, student. That's one very important fact which we are looking into. Very one important fact which we are looking into. Just give me a moment, student. Yes, just give me a moment. Okay. Now, what we are going to see in the next structure, just I have changed the, yes, the background. Let me once know, because the writing pen is not visible. Since the writing pen is not visible, so I am trying to make the writing pen to become visible a little over here. So just give me a little bit of time. Give me a little bit of time. I will just try to come up with the writing pen, student. Let's see if it is possible to get a writing pen right over here. Okay. Let me try to see if I can get a writing pen right over there, student. Okay. If possible, I will get a writing pen. That will be easy for all of us, right? If I get a writing pen, it will be good for all of us. Let me try with that part also. it comes, it will be good for us. We will be able to use that writing pen. Okay. If it is possible, we will try to go with the writing pen, student. That will be really good for all of us. Okay. You will be able to just see the complete structure. Trying to make use of the writing pen, student, just wait. Yes. So that we can go with the writing pen. Okay, let's connect. I hope the writing pen should start working. Yes, we can definitely work with the writing pen, student. Now that is working perfectly. Good, students. Now that is really good. This is really fine, student. Oh, great. Yeah, I guess so. You see, this is the complete structure. I guess this is visible to everyone. Correct, na? Is this visible? All of you? This will be easy for you to this will be easy for all of us to work now. Okay? Let me little zoom in so that now you see, okay, now you see we are going, so we find that there is an anther, there is a pollen grain, pollen sacs are there, anther and filament are present. Now this is better, I can see, yeah, this is now more better. So through these all structures we can easily figure out how the complete structure is going to be working. You see, this, the tip is called as the stigma, then the length is called as the style, the neck, and then we have got the ovary part. In the flower, before I go into the flower, you see this complete anther is a tetrasporangiate condition. This complete anther is a tetrasporangiate where we have got the 4 pollen sacs which are present. The 4 pollen sacs are present, and through this pollen sac, the pollen grains, also we call it as the, okay, these are also called as spore sacs. We also call it as a spore sacs if we use. Now in the complete diagram, if you can easily notice, student, this is how the stigma, growth of the pollen tube is happening right over here. Then we have got the style, we have got the anther, the pollen tube is present, filament is there, male gamete is there, petals, ovules, sepals, female gamete is there, ovary is there, and the thalamus is there. These are some of the structures which you can see. Right, all of you? Everyone is able to understand? Okay, great. Along with that, we also find some more structures. Let's go to the next. Thalamus, as we have discussed before also, thalamus is the base part. Okay, thalamus is the base part, student, and sometimes the thalamus can also grow as a fruit. Sometimes we find that the thalamus can also grow as a fruit, so it can grow as a fruit, and example comes out to be is apple. Apple comes out to be the best example of it. Great, student. Now all the triple fusion, double fertilization, how does the androgen, the growth is going to take place, this all part will do it in the more better detail, do not be worried. As we go a little more further, we will completely discuss this into a much more better detail, student. Shall we go ahead? Okay. So microsporangium structure we are going to do now. Okay, we will go with the microsporangium structure. We will go with the microsporangium structure. And what is this microsporangium structure? How we are going to learn about this? Let us try to do that part also. Now you see there are the layers of epidermis are present. The layers of epidermis will be present right over here, which will be very helpful. Where we have got the endothelium formation, epidermis is there, middle layer is there, microspore mother cell, okay, and tapetal layer. Every layer we are going to learn in much more detail. Before that, there is a little of the complete, that is the important part which we have to discuss, student, which we should definitely discuss. Let me open that part also, important for all of us. Okay, so let us do that particular part also. So what we are going to see is that part is also something which is important, student. Let us do that part also. Yes, students, you should definitely go for. Am I audible to every one of you? Am I audible to every one of you, students? Now, students, you can also go for Unacademy Plus subscription, student. You can also go for Unacademy Plus subscription, and there are many best plan in Karnataka CET 2021. Okay, and if you go for it, like for an example, if you go for a 12-month of plan, okay, a 12-month plan, if you go for, it costs around 12,375 rupees. A 6-month plan will be costing around 10,125 rupees, and a 1-month plan will be charging you around 2,250 rupees. Okay, 2,250 rupees. So this is a trial price only, student. Only for 1 month you get it, for all new subscribers only. So you can just go and you can check how you people are comfortable with the complete, that is the learning through Unacademy, and this will be really helpful for all of you. I can definitely say this will be really helpful for every student over there, and you can definitely feel that yes, it is going to be beneficial for each and every student as such. So that part you can definitely find, student. So that is also something which is really important. So you can go for that also. That is one of the very suggestible kind of a package which you can take, and it will be completely useful for you. Similarly, when you go for this plus subscription, you can find there, student, that there you will get a premium content to be studied over there. Structured live classes will be there, dedicated live doubt clearing sessions are there, quiz at regular intervals, you can see batches for the complete preparation, you can find out. There are batches for the complete preparation, you can find out. That is one very important part. Okay. Then you have got n number of test series, students, n number of test series are there, which you can see, and a single subscription for unlimited access also you can find. Means with a one single subscription, you can go for n number of classes through different faculties what you have. So these all will be helpful for all of you, student. Have you all understood? So these are some of the very important concepts which you have to understand. And over that, there is something which is very important, that is, if you use this code, if you use my code PUNITLIVE, there will be a flat 10% discount, student. there is a flat 10% discount. So there is a flat 10% discount if you use my code, that is PUNITLIVE. You can go for a flat 10% discount, which will be very helpful for all of you. Which will be definitely useful for every one of you students. I hope you have understood. If you can also now follow me, you can also follow me on the YouTube channel, that is NEET UG Made Easy. Okay, where together we can achieve this aim, our goal. Okay, so there is a NEET channel which I am following, that is Punit Sir NEET UG Made Easy and NEET JEE Live. Both are there. Please go and join both of them. Subscribe the channel because you will be getting the classes over there. Okay, as well as you can join me on Instagram, also on Facebook, that is Eklavya Medical Classes, and on Twitter I am there, on LinkedIn I am there. So definitely you can take benefits of there. Also you can see, you can join me over the WhatsApp and you can give me your doubts. You can definitely ask your doubts to me. This is my complete mail ID which I am using, students. So you can come and join me with your doubts on the mail ID also, that you can give it to me. And always remember, student, what is our aim is to get this medical seat. This is the aim, the goal which we are completely working for. This is the aim and goal which we are working for, and I am sure that you people will be able to achieve the aim and goal. And to achieve this aim and goal, student, we have certain success mantra. So what are the success mantra which we have? Okay, the success mantra is very simple. It's very simple and very effective success mantra, students, we are going to have. The success mantra comes out to be is, have a very strong willpower so that you can work hard. Hard working is required. Then have a good dedication, and with a lot of discipline, you people can definitely achieve and aspire the whatever you aim for. You can achieve and aspire whatever you aim for, student. So with this success mantra, anything in your life can be achieved, students. Okay, anything in your life you can achieve, students, with this success mantra. That possibility is definitely over there. Have you all understood, students? So follow this, and it is always going to be beneficial for all of you. Have a very strong willpower, have a hard working capacity, be very dedicated, and be very disciplined to your work. Understood, everyone? Okay. So that's really important, student, for all of us. If we do this, it is definitely going to be helpful for every one of us. Right, student? Shall we go next? Shall we go next, student? Shall we go to the chapter? I think this much we have done, and we should definitely go to the chapter part now. Okay, students, let's go ahead and try to figure out what all we are having. Okay, what all we can do, what all we are having. Let us try to see through the exactly. Let's go ahead. So we have done till here, androecium, gynoecium and all. Now microsporangium structure, this was something which we have not completed yet. The microsporangium structure we have not completed yet. This part we were supposed to be completing. When we go to this microsporangium structure, student, we can easily figure out, we can easily find out. Okay. So, well, you should have, you see, when you go for this complete structure of the anther, anther has got multiple walls where it can be used. Anther has got multiple walls where it can be used and which can be seen in the structures. What are these? One comes out to be is the epidermis, other comes out to be is the endothecium. Okay, I think yeah, some of the students are also chatting with me. So let me look into the chat. What is the recent chat I have got, students, so that I can respond to the students also. Okay. Okay. Let me go ahead with the chats, what the students are trying to communicate with me. Okay. Great. So we'll go next. Okay, let us go to the next. Okay, good. Now, the epidermis of the outermost layer, we can say this is the complete outermost layer. Epidermis serves out to be, epidermis comes out to be a completely outermost structure. So, this is completely the outermost. Okay, I can write it over here. The outermost layer. Outermost layer this is. Okay. When we go to the endothecium, endothecium is inner to the epidermis. So, endothecium comes out to be inner to the epidermis. So, we will write it over here. So, it is inner to the, it is coming out to be inner. Yes, it comes out to be inner to epidermis. It is inner to the epidermis we see. Then we have got the middle layer. The middle layer is multiple layered structure. Even the endothecium is also a multiple layered structure. Okay, middle layer is the multiple layer structure, and also we will have the last layer of, after the middle layer comes out to be the tapetum. Now tapetum, if you see, these are triploid structures. Tapetum, if you see, these are the triploid structure, 3N it is. Okay, 3N triploid structure, and what is the function is they help in the secretion of nourishment. They help in the secretion of the nourishment. They help in secretion of nourishment. They help in the secretion of the nourishment, student, which we are going to see. And through this nourishment only, we find that the complete work is going to be done. The complete working is going to be benefited, and it is going to be done, student. One of the very important factor, one of the very important part which we are going to see. Along with the tapetum, yes, definitely. Yeah, and along with the tapetum, we also find that these are certain cells or cell-like cellular structures are there, which is going to be useful for the tapetum to work. Okay, now secretion to the microspore. Microspore will be present in the center, student. So you see in this diagram, it is very better given. These are the connective tissue. Now, what is the connective tissue, by the way? Connective tissue comes out to be xylem, phloem, which are going to supply the nutrient. These are the xylem, phloem, which is going to supply the nutrient to them, student. And we also find that then comes the epidermis, endothecium is there. Sporogenous tissue, you can see right over here. Sporogenous tissue is also called as spore mother cells. So if I take sporogenous tissue, it is also called as the spore mother cells. We also call them as the spore mother cell, student. A very important fact which we are going to see. We also call it as a spore mother cell. This whole structure will also be called as the spore mother cell. I hope the diagram is much more visible to all of you. So we call it as a spore mother cell, sporogenous tissue, and that leads to the formation of the complete, that is the pollen. Pollen formation is from the spore mother cell only. So you can definitely write over here. So pollen formation. So pollen formation will take place. Pollen formation. Pollen formation is from the sporogenous tissue only. That is one very important factor, which very important part which we are going to look into. Similarly, is the tapetum. The last layer of the middle layer is called as tapetum. Very next to the sporogenous tissue it is present. Very next to the sporogenous tissue. Why am I discussing all this in a more greater detail? Because this will definitely be helpful for you for cracking the KCET exam. That is getting a good rank in the NEET exam. If you get a good rank in the NEET exam, then only you will be getting a good college under the Karnataka entrance exams, right? So KCET, it will be only possible if you are getting a good rank under the NEET exam. So this is a very important part what we are going to see, student. Now this was the completely graphical diagrams which we have seen till now, the layers and how is the complete structure working. If you go for the structure, If you go for the rest of the structure, you find that other notation denotions are also given. Other notations and denotions are also given, which you can easily see. Okay, this can be also easily seen, student, and can easily visible to all of us. So now when we go for the, if you go for a real time diagram, I cannot say it as a real time diagram in fact, but actually if you see under the microscope, it somewhat appears like this only. That is the transverse section of anther, the transverse section of sectioning of the anther. It appears very similar only. You can see how the pollen grains are present. So these are the pollen grains, you can see is scattered. The pollen grains you can find over here is scattered. So these pollen grains are scattered over here, and this scattered pollen grain will be utilized. The scattered pollen grain will be utilized. This pollen will come out of, this is called as a dehiscence point. This is called as the dehiscence point, the point at which the complete, that is the anther ruptures and the pollens are released, and they can be easily transmitted to different places with the help of water, okay, or it can be going with the help of the insects. It can show a hydrophily, it can show anemophily, entomophily, through snail, myrmecophily, through, okay, so malacophily through snail, myrmecophily is through the your ants, it can show, okay, ophidiophily is through reptiles. There is a term which we use, ophidiophily. The ophidiophilo term is used for reptiles, you students, okay, for reptiles we use this term, that is ophidiophily. That is also one very important terms which we are going to use, student. Some of the very common some of the very common terms which are generally used all together, some of the common terms which are generally used all together, which we are going to find. Correct, students? Are you all able to understand? So these are some of the very important terms and terminologies which we are going to use, right? I hope everyone is able to understand properly. If you have any doubt, you can ask me, student. Do not hesitate at all. If you have any doubt, you can definitely ask me for the doubts. Do not at all hesitate for asking the doubt, student. Clear? Good. Now what we next see, what we can next see is, okay, now what we are going to next see over here. So these all parts are done, student. We'll go a little more ahead and we'll see what all part we have in the next slide, coming slides. So let me scroll a little down. Now structure of the pollen, you say, you can beautifully see, student. Now this is the structure of the pollen, what is actually present, okay, that is the pollen grains. You can see over here, see the pollen grains can be of various sizes and shapes, okay, the pollen grains are of various size, various shapes, various structures can be seen in case of the pollen grains. So, but the most important part is, when we are learning about the pollen grains, the most important part which comes into the picture is how the arrangement of the pollen grain has been done, okay, how the arrangement of the pollen grain can be done and how it is going to be present. Now here we find that, you see, this is tetrasporangiate, monosporangiate, bisporangiate, okay, okay, so triosporangiate, different types on the basis of the number of pollen sac which is present in the pollen sacs, which can be noticed, students, which can be seen. So the complete structure of the pollen, you can see that it has got an outermost structure that is called as exine, and after exine they have got the inner layer that is called as entine, okay, the inner layer which we have got, that is called as entine layer, and then inside the entine layer then we'll have the two cells. They are the, one is the generative cell and the vegetative cell. Vegetative cells are a little bigger in size and generative cells are a little smaller in size. So this is the microscopic diagram. I can write it over here so that it will be easy for all of you to understand. So this is the microscopic view diagram. Microscopic view it is. This is also microscopic view, but it has been constructed. You see how the exines are present, student, how nicely the exines are present right over here, you can see, correct, how the exines are present. This orientation is called as, we call it as ornamentation of exine. We use a term, student, that is ornamentation of exine. We use a term that is ornamentation of exine, we generally use for it. And you see how the exines are there. It looks, sometimes it also gives a honeycomb like structure. Honeycomb like structure it gives, okay. So exine is covered with a sporopollenin, student, which reflects UV light and shines bluish in UV light. They reflect the UV light and shines bluish in UV light. So it has got a covering of sporopollenin. Sporopollenin we have got, and through the sporopollenin we find the complete structure to be responding very nicely. It shines very bright in the sunlight. Okay, these are the structure. This is also a microscopic constructed view only. But if we go for a normal, that is the view which is not microscopic but rather it is drawn, you see in this we have got certain these structures like this. Now this will appear like a germ pore. So you see germ pores, many germ pores can be seen over here through which the pollen tube can easily come out. Now these are the points which are called as the dehiscence point. From here we find that the pollens are going to be released. This is the dehiscence point. This point is the dehiscence point, student. This point is the dehiscence point from where it can break off, and we find that the complete structure is going to come out. Okay, what structure is going to come out? That is the pollens, the structure of the pollens. Mature pollens are going to come out of it, student. That is something which is important to be seen and we can understand, student. Have all of you understood this part? Okay. Have all of you understood this concept? Great, student. I hope everyone has understood this concept. Okay, good. Now we need to look into one more part. We need to go into one more part, student, which we are going to see. That is some. Yeah, definitely we are going to learn about that also. Some of the more important parts also are there, which definitely we need to learn, and that is serving it to be some of the importance. So through these structures which we are going to learn, student, what will happen is it is going to be more beneficial for all of us to learn about these parts. Okay, this is going to be more beneficial for all of us. Shall we start now? So I'll start with this. Let us go ahead, student. Till here, is it clear to every one of you? I hope till here it is clear to all of you, students. Everyone has understood till here? See how beautifully these are designed. You can see how beautifully these are designed, student, as you can see. Correct, huh? Shall I go a little more or less? Next. So you see a very beautiful structure of the pollen, as you can see over here. One of the very beautiful structure of the pollen, what we can right now seeing over here. How good it looks like. Germ pores and all, we are going to see some more structures which are important. Germ pores and all, we are going to see some of the more structures, student, which will make the complete learning a little more interactive, and you will be able to understand the structure proper, right? Okay, let's go ahead. Structure of the pollen, you see. When we go to the structure of the pollen, student, now we are continuing with the structure of the pollen. Shall we start now? Yeah, we'll go with the structure of the pollen right now, and we'll see how the structure of the pollen is going to be helpful and how it's going to be working. Shall we? Okay. So let's go with the ahead with the structure of the pollen now. Let's continue with the structure of the pollen. You see, the pollen structure completely it has got the two cells, that is the we have got the complete tube nucleus right over here, then we have got the cytoplasm, the germ pore. I'll discuss everything in more detail. Exine, intine is there, nucleus is there, cytoplasm is there, and generative cells are there, as well as we have got the generative cells also, right students? So when we go to the tube nucleus, we also find that the complete cytoplasm, the tube nucleus is coming from the complete vegetative cells. Okay, the tube nucleus comes from the vegetative cells. Then we have got the cytoplasm, which will be nourishing the structure completely. Okay, which will be nourishing the structures completely. Then we have got the germ pore. Germ pore is the place from where we find that, you see, exine is absent, only intines are present. So germ pore is the structure where we find that only intines are present and exines are generally absent in these regions. And you see the exine is completely not present over there, intines are completely going to be present over there, and exines are absent. So one more germ pore I can see right over here. So if there are two germ pore and two pollen tubes are coming, if there is a two germ pore and two pollen tube are coming, we call it as a disymphony. We call it as a disymphony. Am I clear? We call it as a disymphony. Now you see how beautifully they are given. The tube nucleus will be forming. The tube nucleus will be forming the pollen tube. That will lead to the formation of the pollen tube. Now this is the complete structure of the pollen tube, as you can see over here. This is coming downward. Okay, the pollen tubes are coming down, and we can find that the generative cell has divided into two male nuclei, and this two male nuclei is developed by mitosis. That is important. The two tube nuclei are developed by mitosis, student. They are going to be developed by mitosis. By mitosis. To be developed is mitosis. Understood? It's coming through the mitosis. Okay. Now what do we see? That the pollen grain is there, aperture is there, and the exine is there. We find the pollen tube. Now there are, you see from here the three pollen tubes can come out, and since the three pollen tube can come out from here, we call it as the three pollen tubes, so we call it as polysymphony. We call it as polysymphony. Okay. And if one pollen tube is coming out, it is called as monosymphony. Two coming out is called as a disymphony, student. We call it as a disymphony. If the two are coming out, we call it as a disymphony. That structure will be called as a disymphony. So disymphony, monosymphony, polysymphony is there. These kind of structures can be seen. Have you all understood till here? I hope for everyone it is clear, students. Till here, is it clear? Now we have got the sporophyll and the gynoecium. Sporophyll to the gynoecium, we have got how the formation is going to take place. Okay, you see how does the development of the carpel is going to take place, what we can find out. Okay, You just give me a moment, student. I hope the concept is understood to every one of you now. Correct? Okay. Now these are the leaves which, how is the carpel or a gynoecium is going to be formed, that we are going to learn now, students. How the formation is going to happen, that is the leaves. Actually the modification of the anther or the pollen is nothing but it is occurring through the complete, that is the androecium, through the leaves only. Through the leaves only we find that this complete structure is going to be developed. Androecium and gynoecium, they are the product. They are said to be the product of the modification of leaf which we are going to see, student. They are all the modification of the leaves what we are going to find out, student. That is one of the very important part which we are going to see. Have you all understood? I hope everyone is able to understand that. Okay. So what we are going to see now. Okay. So what we are going to see now is, you see how the leaf they roll on. The leaves are going to be rolling on, and that lead to the production of the complete structure. So the leaf is going to be rolled on. So the rolling is taking place in this manner, and that leads to the formation of the complete androecium and gynoecium. That formation is going to take place, student, as we can see over here. I hope you are all able to understand this part. Correct? Now we also find that in case of anther also, you see that is the folding of the leaves only which are going to take place. The anther also, the folding of the leaves are only going to take place, and this leads to the formation of the complete, that is the carpel. Okay. So tricarpellary pistil, you see it is 3 carpels are there. Tricarpellary pistil or gynoecium we can see over here. It is a tricarpellary structure. Okay, student. So that is one very important factor which we are going to see. One pistil is there, 3 styles will be present. One pistil with the 3 styles are present, student. That is one very important part which you are going to look into. Okay, trilocular we say also, 3 locules can be seen over here. Okay, so it can also be called to be a trilocular. Let me change the color. So also we call it as a trilocular structure. This is a trilocular structure what we are going to see here, student. Okay, 3 locules can be seen separately. So these are the how we have got the trilocular structure. Have you all understood till here? Is it clear to every one of you, students? Okay, let's go to the other part, next part of it, and we'll try to see through those structures. So when we go to the types of gynoecium, you can easily figure out the types of gynoecium can be of different types. One is called to be apocarpy. Okay, then we have got the pericarpy, and locules are also present. Apocarpy, pericarpy, as well as the locules are also present. Locula, so monolocular, bilocular, trilocular, tetralocular, all such structures can be seen. All such structures can be easily noticed and can be seen, student. So we have got the apocarpy, pericarpy, Okay, what is apocarpus? You see, apocarpus condition, pericarpus, apocarpus condition, we see the outermost layer covering is there, pericarpus. Okay, then inside we have got the formation of the locule. In the locule, this is the septum through which the division of the complete locule is going to be. These are the septums, and in the septum we have got the formation of the ovule. In the septum we have got the formation of the ovule, student, which can be easily seen. In the septum we have got the formation of the locule, which can be easily seen. This is a 3D structure. They are trying to show a trilocular ovule. How is a trilocular ovule present? This is a structure they are trying to show. Can you see? So right over here we can see a trilocular structure is there. Trilocular structure is there. You see there are 3 locules which are present, students, right? Is it visible to every one of you? And this is how the septum is going to divide the locule very properly. Syncarpus is a condition where we find that the complete locule is going to be fused. All the ovules, when they are going to fuse with each other, that is particularly called as the syncarpus condition. When the locules are going to be fused with each other, we call it as a complete, that is the syncarpus condition, student. We call it as a syncarpus condition when the locules are going to be fusing with each other. One very important part which we are going to see, student. Okay, have you all understood? I hope with the diagram it is more visible and is more understood to every one of you. You can find that. Correct, student? So these are some of the very important structures which we are going to see. These are some of the very important structures which we are going to see, student. I hope everyone is able to understand with these structures. Correct? So I think it is completely understood how it is going to work out and how it is going to be completely structured together, layered together and structured together. Right, student? I hope everyone is able to understand the concept. Correct? Correct, my students? Okay. Now we see the structure of the megasporangia or the ovule. Let us go with the structure of the megasporangia or the ovule structure. You see, we have got a structure that is called as raphe, R-A-P-H-A-E. Then we have got the hilum. Hilum is present. Chalaza is there. Nucleus is there. Antipodals are also present, students. Okay. We will learn about the embryo sac, then secondary nucleus, egg cells, synergids. All together we are going to learn about it. Let's go ahead and try to figure out how the structure is going to be formed. Let's see. Raphe is a structure. Raphe is a structure which is going to attach the complete ovule to the funicle. So I can say this is the attachment point of the complete ovule to the funicle. This complete attachment is with the help of the raphe only, which is going to happen. That's one thing. Then comes out to be is the hilum. Hilum is a place. So hilum comes out to be a place from where the funicle arises. I can say hilum is a place from where the funicle arises. So from here I can say funicle arises. Funicle arises. Then we have got the inner integuments and the outer integuments, student. Then we have got the inner integuments. Okay. Then we have got the outer integuments, that also we see. Similarly, we have some more structures. Okay, similarly, we have got some more structure, we call it as a micropipe. So, what is the inner and the outer integuments? Let's try to understand. You see, students, this is the outer integument, this is the inner integument. This is the outer integument, this is the inner integument. They protect the complete ovule, students. They are going to completely protect the ovule and they will be helpful for the movement of the complete, that is the pollen tube, how it is going to move, the pollen tube movement, how it is happening. we are going to learn with the help of the integument structures. The structure of the integument only we are going to be helpful to be understood. Now when we go to the ovule, we have a central nucleus, that is what we are going to see over here. So initially we have got the central nucleus, and the central nucleus is diploid. Let me tell you, it has two nuclei. Okay, so it is two nucleus, haploid, two nucleus, I can say like that. Okay, it's not a diploid condition, it's a haploid, two nucleus are present. So n plus n, I can say definitely, not 2n. We can say rather n plus n structure is present right over here. Then we have got the antipodals. Antipodals are all n, haploid. Synergies are also haploid, and egg cell definitely has to be haploid only, n is present. So synergies, they protect the egg cell. During the penetration of the pollen tube, they are going to protect the egg cell, so that while the pollen tube is entering into the egg, it should not be jerky, it should not be destroying the egg of the complete ovule. That is very important, student. Now the secondary nucleus, which is n plus n condition, what is the use of the secondary nucleus is, it helps in the formation of the endosperm. So when one male gamete will go and fuse with it, it will form a triploid, that is 3n structure. After fertilization, a 3n structure will be formed, which will be nothing but the endosperm. Endosperm formation will take place. Similarly, antipodals are n, haploid condition. Antipodals are said to be n, that is the haploid condition, it is going to be over there. So these are some of the structures which you are going to see, students. And the uppermost part is called as the chalazal end, and this is called as the micropyle end or micropylar region. So upper part is called as a chalazal end, and the lower part is called as a micropyle region. The upper part is called as the chalazal end, and the lower part is called as the micropylar region. This is the micropylar region, and the upper end is called as the chalazal end. Am I clear, students? Have you all understood till here? Okay. So these are some of the structures which we are going to see, which we are going to notice, students. I hope till here everyone has understood the concept. I hope till here everyone has understood the concept, student. Right? Okay, good. Then there is a terminology which you are going to use, that is called as microsporogenesis. Okay, there is a terminology which we generally use as a microsporogenesis, student. Just give me a moment. Okay, now we are going to learn about a little about the microsporogenesis, student. So when we go to the microsporogenesis, or how the formation of the megaspore is going to take place, the megasporogenesis formation of the ovule, this part we are going to learn a little bit and will understand. Okay. Now you see, from the micropylar end, there is the cell growth taking place, that is called as nucellus. Okay, the cell growth is going to take place, that is called as nucellus. As well as the megaspore mother cell. The megaspore mother cell is also there, or a spore mother cell. We also call it as spore mother cell. SMCs also we call it as. SMCs, that is the spore mother cell we call it as. And we generally see that from the micropylar end the division starts. It leads to the formation of a megaspore dyad. Okay, the nucellus divides and leads to the formation of a megaspore dyad. As the megaspore dyad is going to form, student, as the megaspore dyad is going to take place, student, in that we find that at the micropylar end we are going to have, at the micropylar end we can easily find that we are going to have the complete, that is the tetrad formation, 4 layered structure, a tetrad formation is going to take place. Okay, and this is also called as a megaspore tetrad structure. And from the chalazal end and the antipodal end, as well as the micropylar end. Okay. Now this leads to the formation of the complete tetrad, out of which 3 are going to die. In the tetrad we will find that the 3 cells will die, and only one is going to survive. Only one cell is going to survive, student. Only one cell is going to survive, student. Am I audible to every one of you? And rest we find that as the division starts taking place, then the one cell which has survived will lead to the formation of the complete ovule. So next all division which is going to take place will be mitotic division, because it is already haploid condition. So it will lead to the formation of mitotic division. It is going to take place. Now what is going to take place? 2 cell, 4 cell and 8 cell stage. So we say 8 nucleus stage. So we say that it is a 7 celled 8 nucleate structure. Nucleate structure. 7 celled 8 nucleate structure we call it as, where we have got the chalazal end, where we have got the antipodals, then we have got the polar nuclei, okay, with the XL, synergids and filiform apparatus which appears on the synergids. So these are the filiform apparatus which are appearing on the synergids, what we can see, students. They are appearing on the synergids, what we can see. Have you all understood? So this structure comes out to be one of the very important structure, students, which can be seen. The structure comes out to be one of the very important structure, student, which we are going to see, and it will be very much contributing towards the complete spore development. Okay, is going to be helping in the complete spore development. I hope all of you are able to understand. I hope all of you are able to understand, right? Correct, students? Have you all understood this part? Okay. So these are the complete structure of the megasporangia. Let us do it in a little more detail regarding the theory part. That is, okay, let's do it in a more theory part. That is, the embryo sac are usually 8 nucleate and 7 cell structure. Type of embryo you see, can be a monosporic, that is the polygonum type, a bisporic, that is Allium cepa, tetrasporic, that is the adoxa type. Allium cepa is nothing but is your onion. Okay, that is how we have got. Then we have got adoxa. More than 8 cell in the embryo, if you see, more than 8 cell in the embryo, if you notice, then the cells are peperomia type, pinnae type, drusa type, fritillaria type. So these are more than 8 cell in the embryo sac. If we have, then we call it as, okay, peperomia type, pinnae type, drusa type and fritillaria type. These are some important points, student. These are asked in AIMS, JIPMER and AFMC. That are all things which we are going to see, students. They are going to come in this particular way. We can completely understand this part, student. Have you all understood? Every one of you? That is what we see. A structure of megasporangia. When we see the structure of the megasporangia or the ovule, the integument can be unitegmic. Only one integument is present. Unitegmic, we are going to find that only one integument is present. You see over here in case of Cycas, Pinus, Solanaceae family and Compositae family. Okay. Yes. Okay. Now we find that Cycas and Pinus species all are gymnosperms. In gymnosperm type, we are going to see Solanaceae is the family of vegetable, and Compositae you see Tagetes, then yes, that is the marigold. Then we have got sunflower, Helianthus. Okay, we can use the term Helianthus. So these are some of the terminology we are going to use, students. Helianthus, we find such a structure. Okay, these are some of very important terms which we have till now understood, students. I hope this is clear and this is understood to every one of you. correct? That is what you see, student. Okay. Next, what we are going to look into, okay. So by technique, if you see, then we have got crucifery. There is the two integuments are present, two integuments. There is in crucifery. Mustard comes under that, okay. That is the cross shaped flowers can be seen. Petals are cross shaped, so we call it as crucifery. What is the crucifery? Mustard? What is the name which we are going to use for mustard? What is the botanical name which we use for mustard? Biological nomenclature, how it is done for mustard? How do we do it? Yes, Brassica campestris. Brassica campestris. We use A technique is without integument. Lawrence family, then Santalaceae family. Lawrence Santalaceae family, we find that it is absolutely going to be A technique. So unitegmic, bitegmic, ategmic, in this way we have got the complete structure distribution, and this is how we have got the complete, that is the distribution of the integuments in case of the such a structure. I hope this still here it is clear to every one of you. I hope this is clear to every one of you. Okay. Now we will go to the summary what we have learned today. So today we have studied about the flowers, then microsporangium structure, structure of the pollen, structure of the megasporangia, and microsporogenesis. These all part we have learned today. Now what we see is, students, you can also definitely go for the plus courses of Unacademy, which is going to be very useful for all of you. Okay. And it is really beneficial for the students who are preparing for the NEET exams. Why it is, let us see. So plus subscription you can definitely go and opt for best plan in the Karnataka we have got, which is a very low amount, that is 12,375 rupees. It is 6-month plan also you can go for, which is 10,125, but I would rather suggest you to go for the 12,000 only, because there is hardly a difference of 2,000 something rupees which is going to take place, which will be definitely useful for every one of you. So I would rather suggest you to go for a 6-month plan only, and you see 1-month plan you can go, which is just for a new subscription. If you take, then only you can go for a 1-month plan, and it is just for a trial basis, and I'll definitely, you people will like the content. What you are going to get under this will be really good for all of you. Okay. So like premium content at affordable price will be given, structured live courses will be given, quizzes at regular interval will be given. Okay, batches will be there for completion, test series and DPPs will be there, single subscription for the unlimited access will be there, student. Always remember, students, our goal and our success mantra, that is the willpower. You have to be having a high willpower, a good willpower, strong willpower, hard work, dedication, and discipline has to be there in your life. Then only you can achieve the success in your life, student. Very important. If you use this code, if you use my code, student, while taking any live subscription, definitely you get a discount of 10%. So a flat 10% of discount is there if you use this code. So definitely you can use this code, students, where you get a 10% of discount, student. Definitely it will be useful for all of you. Have you all understood, students, this part? Has everyone understood this part, student? I hope everyone has understood with this concept. So good, student, we'll meet in the next class and we'll try to learn little more through the live classes. That's really great, student. Okay. By the meantime, yes, thank you all of you. Yes, by the meantime, we'll I'll just say thank you all of you, and we'll meet in the next class, students. Okay? I guess all of you have understood this part and this concept. Correct? Shall we go ahead?